Okay, we all got the recording announcement. Terrific. So I'm the Chief Program Officer at MHAMD, and in my role, I have the privilege of overseeing 4 program areas, one of which is mental health first aid. We also have a children's mental health division, an older adults mental health division, and also a consumer quality team, which visits psychiatric care facilities across the state of Maryland to make sure that patients are getting the quality of care that they want and need. So in the next few minutes, I'm probably going to take 5 to 8 minutes max, just to give you a little bit of context and where we're going and where we've been the past year in mental health first aid. Just quickly, a little bit about myself personally. I joined the association in November of this year. I've spent the past 30 years in mental health in different areas, including practice, administration, research, and public education. I was really excited to take this position at the association because it returned me to mental health first aid. And I'm looking in the audience to see if there's anyone who I worked with when I was at the council, kind of scanning names. But I came to Maryland after being at the council for a number of years. And so I'm really excited about this program. And before I go on, I want to stop for 2 seconds and thank the team. I know all of you know Anne, and Anne has been a constant steward of this program during a lot of upheaval. So Anne has the most sunny disposition of any professional you'll work with. So cheers to Anne. And she has been joined by Leah Bentfield, who has been an incredible add to our team. Leah is kind of heading up our social media and also looking at our business process. So she will probably be interfacing with a lot of you over time. So Leah, thank you for all that you've done. And I hope that we'll see kind of the neat things that we're even doing in this virtual instructor summit that she has brought our way. Brian Gibb, who's also on the call. Brian, another thank you to Brian, who's on our team. Most of you probably have worked with Brian or know Brian also from his long tenure at the council, I think over a decade, in building mental health first aid. So he too shares great excitement for this program. So thank you to the internal team, which is not the purpose of this meeting. The purpose of this meeting is to thank the external team. So we want to thank all of you for all of the hard work that you've done this past year. The past 15 months, I'm sure all of us have a different word for it, but we know it's been challenging. And despite how challenging it's been, we have gone and done incredible things in our communities and in our programs. So I just wanted to say that I recognize how hard COVID-19 has been for all of us, for the world, for our work, for our families, for some of our own personal health, and also just for the instruction of mental health first aid. All of you have joined together to be incredibly flexible and nimble and committed. And I say that because it's not just the commitment of the heart, it's also the commitment of the action. And we are really fortunate to share with you that we've had some great training numbers in the past 15 months, actually in the past year. And so I share with you that you as instructors have increased the community trainings throughout the state by over 45% during a pandemic. That's awesome. It's incredible. And also to my small and mighty team that we work together with, we have increased instructor trainings during this pandemic also by 50%. So there has been a tremendous amount of work, not just in managing the technology, moving to virtual, keeping people engaged, but also to responding to our communities. And I think all of us are here today because we care deeply about the field of mental health. And post-pandemic and during the pandemic, we know there has never been more of a need in our communities for this particular program. So I'm sure that as you've taught this year, it's been incredibly challenging and rewarding. So I thank you for all the time and all the effort that you've put into this program. A couple things about what have we done and what's coming up, just so that we can give you some announcements programmatically. We are intending to have a lot of growth. We had a lot of growth last year, and we're intending to have a lot more growth. And so about 1% of Marylanders have been trained in mental health first aid. Well, 1% is still a lot of people, although we really have goals to increase that percentage. It is really no different than the rest of the country who's been ambitious in mental health first aid. So we are doing a great job. We just know that we need to get this training out to more and more professionals over time. And so the growth is our number one goal. And in order to do that, we have added new, we actually doubled our capacity for Maryland national trainers, our Maryland trainers. So a shout out to everybody who's agreed to take on that audacious job. And so we're building our capacity internally so that we can train more instructors and get this program out. In terms of instructor support, I think whether you're seasoned and have been doing this 10 years, 2 months, we all need support. It's hard work. And we need support with instruction. We need support with our networking. And so our commitment to you in the state of Maryland is to do a better job with instructor support. We'll be offering additional webinars. We will be offering additional regional meetings. We will be doing a better job at reaching out to you and making sure we're meeting your needs as instructors in the coming year. I hope some of you have participated in our webinar series that we partnered with the Dania Institute last spring. Anybody do that? Head nod, yeah? Okay. The great thing that we hope to do is really start partnering with content experts to bring additional resources to our communities in terms of our instructor community. So we look forward, and I hope next year we will not be on Zoom. I hope we'll have an instructor summit where we have lunch together, we share, we celebrate, we get to actually see each other. So that's a little bit in terms of instructor support. Quality, I want to share real quickly that quality above all else. You can have all the reach in the world, but if your quality suffers, it's not worth doing, right? So we are strongly committed to quality, and we have just put in place this spring a quality assurance program for our Maryland trainers. And so we will make sure that those trainers who train instructors are getting ongoing support, coaching. We're learning from them, they're learning from us. They're stellar in what they do. And we know that this constant back and forth of quality will be really impactful. Curriculum, I want to jump to that. I'm going to try to be as brief as I can. Many of you have probably heard about the teen mental health first aid program that was piloted by the council throughout the nation, and they've had some incredible results. We at Maryland are working hard to acquire that curriculum. We intend to have that curriculum this year, hopefully before the calendar year ends. And that is an incredible opportunity for us. I think all of you who work with youth know that this, whether it's pandemic or post, we need this curriculum. And there has been a research study done by the Harris, the National 4-H and the Harris poll that showed during the pandemic, 7 out of 10 of our high school students are struggling with stress, depression, anxiety, or all three. So this is going to be a wonderful opportunity to bring teen mental health to our school systems. And it doesn't go alone. It travels with training the adults in the building on youth mental health first aid. So great program. We're really excited to be able to offer it to you shortly. And so please stay forward, look out for updates from us. It's only for students in grades 10 through 12. It's taught at a grade level at a time. So there's real impact and sustainability in a school building. Students still learn a 5-point action plan. The most important thing they do is reach out and get a trusted adult. So anyways, more to come on that. Real quick on rebranding, those of you who are at NatCon or work with the National Council know that they have changed their name, their look. They are now the National Council for Mental Wellbeing from the National Council for Behavioral Health. And their logos look a little different, but they're still doing the same kind of work, but just positioning a little bit to look at prevention and wellness as well. And so what does that mean to Maryland? It really means that we are going to try to align with their branding in the coming months. Nothing will change as far as you can use all of your closets full of materials, whatever way they're branded, anything that you have, but eventually we will align our branding so that folks who want to take the training are not confused in any way. And last, I just wanted to say that in order to support all of the growth that we want to see in this program, we've been fortunate to have received some additional funding from the state of Maryland Behavioral Health Association, which is terrific. We have not had new funding in a long time. So they are recognizing the needs in our communities. So that's great. We'll support that. And at the present moment, we will keep you all posted on whether or not what we're doing as far as our own remote work and instruction. So that's the update a little bit from the council. Kudos to all of you for all the great work you're doing, for all the flexibility you've shown during this pandemic, for your commitment, for being here during the lunch hour. And I'm not even seeing anybody eating, so please eat if you need to. But anyways, I'm grateful to having had the opportunity to be here, and best of luck in the coming year. And I'm going to pass it back to Anne.
Thank you, Mary. I just want to reiterate Mary's point, which is that you instructors are the heart of Mental Health First Aid Maryland. And we are so grateful to each of you. We want to support you in all of your work. We want to celebrate you. And part of what today's program is, we want to give you more tools to do what you're already doing so well. So Mary's going to jump off. She has another meeting, I believe. Thank you. And we are going to introduce Dr. Paul Shepard, who is our keynote speaker today. Dr. Shepard is a professor of psychiatry and pharmacology at the University of Maryland School of Medicine. He holds a PhD in medical physiology from the University of Texas. He has a postdoctoral degree from Yale University School of Medicine in neuropsychopharmacology. Dr. Shepard has spent nearly 30 years researching dopamine and its role in brain diseases and then treatment of diseases of the brain. Dr. Shepard is going to talk about some basic principles of neuroscience and then connect them to some of the concepts that we as mental health first aid instructors teach in our curriculum. As we listen to Dr. Shepard's remarks, I want you to have your mental health first aid instructor hat on the entire time and think about some of these core tenets. We don't diagnose. Dr. Shepard's going to talk to us today about insights into the brain that will help us to understand some of those concepts that underlie our curricula. We are noticers. Dr. Shepard's presentation is going to help us hone our noticing skills, especially skills around behaviors. We connect people to care. We'll be talking about, Dr. Shepard will be talking about some recent advances in treatment options. And then that incredibly important hope discussion. We infuse hope into the conversation around mental health where it has historically been missing. There has never been a better time to be talking about hope, as exciting advances in the field are happening all the time. And Dr. Shepard's going to talk about some of those as well. When we connect people to the appropriate care, recovery is the expected outcome. It is not just hoped for, we expect recovery. So all of us, as we go forth with our mental health first aid message, are able to really infuse our discussion with tools and reality and insights into all the exciting possibilities. There has never been a better time to talk about treatment and recovery from mental health challenges. And with that, I'm going to hand the digital baton to Dr. Shepard.
And that was very kind. Thank you so much. Let's get past the technical part of this and see if I can successfully share the part of my screen that's important. Does that look right to everyone? It looks good at this end. Let me move people around here a bit. There we are. Okay, so you guys should be seeing a blue screen with a title of the talk and my name at the bottom of it. Is that what you're seeing?
Looks good.
Fantastic. So let me tell you what a great pleasure it is for me to be here to share some concepts around basic neuroscience principles and how they relate to serious mental illness. As Ann said, I've spent most of my career thinking about this, mainly as a preclinical scientist. I don't want to leave anyone with the impression that I am a physician. I certainly am not. Ann and I met actually in the, I was fortunate enough to have the opportunity to take the adult first aid course in September of last year. And Ann and I got to talking as part of that course about some of the material and how it might relate to concepts that you guys talk about and really implement. So a dialogue developed between the instructors and between the scientists in the group. And that's really how this talk sort of came to pass. To be honest, and I really, just and I don't mean to, I just want to make sure that you guys understand, I don't know how much of what I present today will actually be of any practical use to you as instructors. What I hope is that the material is going to stimulate some curiosity on your part to delve deeper into the basic brain mechanisms that underlie serious mental illness. And there's one in particular, as Ann mentioned, that we're very excited about. And I'll discuss that briefly at the end of the talk. So as Ann said, and like you guys, neuroscientists are really don't diagnose. We're trained to notice things that happen in the brain. And speaking as a preclinical scientist, I can say that if we're wildly successful in our careers, and very few of us really are, the beginning of a new treatment is developed. In reality, most of the therapeutics that are used to treat serious mental illness have been discovered serendipitously, and we're going to again end the talk with a really fine example of that. So more often than not, at least the payoff from our work is far less tangible. It does, however, result in an increased understanding of how the brain works. And that's a cumulative process, right? So we stand on each other's shoulders, and that's some of what I'm going to be sharing with you today. So we'll begin with some structural features of the brain. Hopefully you guys can see my pointer. We'll talk a little bit about gross anatomy. I'm not going to say too much about gray versus white matter, but we're going to delve rather into the neuron. This is sort of the linchpin of what we'll be talking about. And we'll be discussing separately the three components that make up neurons: dendrites, axons, and nerve terminals. And then after that, we'll talk a little bit about the whole concept of neurotransmission, how neurotransmitters are released, what they interact with postsynaptically. And after about 15 minutes of reviewing stuff like that, we're going to start to delve into some neuroscience concepts. And as Ann mentioned, we'll be talking about drug abuse. I know you guys are interested in how naloxone is working in humans. We're going to review all of that at sort of a molecular level. We'll talk about this concept of plasticity, which is really central not only to the normal function of the brain, but also disease, and then conclude the talk with a little bit of the neurobiology of stress and how it relates to panic disorder and PTSD. So the fact that we're all together here, I think this morning is evidence that we share some fascination with the human brain. I mean, along with performing millions of little mundane tasks that we all do during our lives, it's capable of things like composing concertos and coming up with solutions to equations. It's really quite an amazing, it's an amazing thing. The brain in this Koppen jar weighs about as much as a small sack of potatoes, about 3 pounds typically, and yet it's comprised of nearly 100 billion individual neurons. And it's no surprise maybe as a result of that hyperbole can sometimes make its way into discussions about the human brain. One of the misconceptions that's pretty popular here fairly often is that we use only about 10% of our brains, which is a complete misnomer. The fact is that our brains have evolved to work extremely efficiently. And that doesn't mean that we're not using them most of the time. And in fact, some tangible evidence of that is that while humans, the human brain only accounts for about 2% of our total body mass, it consumes 20% of the total energy budget, our total energy budget. It's pretty remarkable. So we're going to start with a little bit of gross anatomy in the brain, and the brain has some functional regions, as you might imagine, and for the purpose of today's talk, I've just combined them into 3. So to orient you, this is the front of the brain, and what we've done is we've just simply run a scalpel straight down the central fissure and broken the brain into 2 symmetrical halves. So what we're looking at is sort of from the inside of that little split that we just made, the structures that you would see if you just divided the brain in half along its longitudinal axis. So here's the back of the brain. There's the top of it, and here's the front of it. So the hindbrain, which is this part in blue, is clearly the most primitive. It has the lowest density of neurons, this part of the brain, which is arguably the most important part because it's keeping us breathing and keeping our hearts beating. Has the fewest, the lowest density of neurons, but has this very important role of being the origin of cranial nerves, of relaying sensory information from the spinal cord up to the more advanced or front parts of the brain. It's responsible for nearly all of the autonomic functions, keeping our heart beating and us breathing. And this small part of this sort of mini brain here, the cerebellum, it's such a peculiar place. We still don't know much about it. It certainly is involved in the regulation of balance and in the origins of motor movement. It has the highest density of neurons in any part of the brain. They're just jam-packed in there. And it's an area that we need to make more progress in. We don't know much about. There's this little red area here, which is my favorite part of the brain. It contains the neurotransmitter or cells that use the neurotransmitter dopamine. These, of course, are the cells that die off in Parkinson's disease, and also the VTA, which is another dopaminergic area that's involved in drug abuse. And we'll talk about that some more in just a minute. And then this entire yellow part of the brain, which is really comprised of two large parts. The telencephalon, which of course accounts for most of the cortex, and that has several different lobes. The frontal lobe is really the most advanced part of the human brain. It contains neurons that are engaged in the business of controlling muscle movement, decision-making, thinking, planning. The parietal cortex is mainly involved in processing of sensory information coming up through the spinal cord and this primitive area that I was telling you about. And the occipital part of the cerebral cortex is involved in vision. The temporal cortex is an unusual place. It's where most of the limbic system is located. The amygdala lives here, and so does the hippocampus. And we'll be talking about those brain areas in the context of panic and stress-related disorders in just a minute. So beneath these cortical areas is a region that is not highlighted here, but is extremely important in the normal functioning of the brain. It contains the thalamus and the hypothalamus. So the thalamus, of course, is involved in coordinating all of that sensory information on its way up to the frontal cortex. A lot of it also passes through the thalamus on the way down. So people tend to describe it, if you're looking for the words, or like a brain's conductor. And the hypothalamus, of course, is an important part of the neuroendocrine part of the brain that's involved in the control of hormones and intimately involved, not surprisingly, in stress. So each of these brain areas is comprised of neurons, as you already know. Neuronal shape varies widely within brain regions and defines, in many respects, the kind of processing that occurs. But for the most part, we're going to, for today's talk for sure, we're going to ignore all that diversity, and we're just going to focus in on the three principal areas of neurons: the dendritic fields, which come off of the cell body, this long part, the axon, and then this region here, the axon terminals, where neurotransmitter release occurs. We're also going to be focusing in on synapses, and really, their synapses are worthy of special mention because they pass and they shape the information between neurons. Their function is to change chemical energy into electrical energy and back into chemical energy again. It's really quite an amazing transduction mechanism. We'll look into that not only as just a core concept, but as a site for where drugs act. Synapses are really where most of the therapeutic strategies that are developed by basic neuroscientists are targeted. And there are a bunch of them in the brain. I mean, if you do the math, imagine one neuron, and there are, what did we say, 85 billion of them in the brain, and each neuron is capable of forming somewhere around, if you were to make sort of a ridiculous average, of around 10,000 synapses. So you can imagine how many of these things are in your brain and how many of them are active at any particular time. It's just it's mind blowing. So let's start with the input side of the neuron. This is so the way I think the way to think about how the nervous system is put together, and particularly in the context of pathways, is that neurons are, of course, are connected together in chains, right? And it all starts with the this first part, the dendritic tree. We call it a tree. It extends from the cell body and with these fibrous like things. And if we zoom into one of them, they're not smooth as they appear here, but they're reticulated with these little spines, these tiny little blebs. And the spines are points of contact with other neurons, which is an important thing. So if we were to now, of course, when a synapse is activated, the thing doesn't move like it's moving here. I'm using this to simulate the fact that the synapse is activated. And what's happening, of course, is neurotransmitter is being released from each of these. We're going to talk about this in a minute. Something's just happened. There we go. So neurotransmitters coming out. And when that happens, a small electrical potential is generated postsynaptically on this neuron that is the target of this afferent, a local electrical potential. And these potentials travel passively as a result of the sort of the structure and how these membranes are put together, travels up towards the cell body. And if you can imagine these things being generated all over the dendritic tree at any given time, this at one level, this is how information in the nervous system is integrated. You have these potentials. Some of them are inhibitory, some of them are excitatory, and they're being conveyed and bombarding these dendritic fields on neurons all throughout the brain. Even when we're sleeping, this is occurring. So what happens when they get down to the cell body? Well, of course, they continue to propagate, and they move across the cell body to a very specialized region of the axon called the axon hillock. And this is a lowest resistance area of the neuron. And when all of these inhibitory and excitatory potentials meet here, they generate an action potential. And this action potential then courses down the axon towards the nerve terminal, depolarizing the nerve terminal and causing neurotransmitter release. So let's pick that up on this diagram, which is sort of a little different. So here's our dendritic field. This is where all of our neurotransmitters have been bombarding these postsynaptic receptors on the dendritic fields. And as we'll learn, some neurotransmitters are inhibitory, some are excitatory. So there's a very important level of integration that occurs here in the dendritic fields, right? So there we go again. A very important level of integration that occurs here, but yet an action potential is generated or it's not, right? And that courses down this axon. And if you saw it, it's invading this nerve terminal. And what happens as a result of that is these little bags of neurotransmitter are literally physically moving. So this is one instance when the cartoon is really right. There is a physical translocation of these bags of neurotransmitters called synaptic vesicles to the tip of this nerve terminal, and the membrane fuses here and the neurotransmitter spills out into this small space called the synaptic cleft, where the neurotransmitters are then free to interact with postsynaptic neurons. So at one level, the language of neurotransmission is in the form of the neurotransmitters that the brain is capable of making, right? And there are a bunch of them. There are around 60 different types of neurotransmitters in the brain. People argue about what constitutes a neurotransmitter, but there are probably 60 or so in total. Some of them are amino acids like glutamate. We've heard of glutamate and GABA. And those of you that have some familiarity with neurotransmitters and their function in the brain will know that glutamate is almost always excitatory. And GABA is almost always inhibitory. So think of that diagram where we started, where these neurotransmitters are sort of coming out of these terminals, and sometimes they're hyperpolarizing the neuron, making it less excitable. Sometimes they're depolarizing it, making it more excitable. These little inhibitory and excitatory postsynaptic potentials, as we call them, are coursing across the cell bodies towards the initial segment of the axon. And then an action potential is either generated or it's not. That really is sort of the core level of integration in the nervous system. And you can imagine, given the number of neurons in the brain, at what scope this is happening in any minute. So we have a bunch of different types of neurotransmitters. In addition to these simple amino acid ones, we have some like dopamine, serotonin, and norepinephrine, which are more complicated. And as we'll see in a minute when we talk about receptors, they do different things in the brain than these fast excitatory neurotransmitters. Acetylcholine, of course, is used in the nervous system. Not as much of it in quantity, and in fact, by complete accident, I've listed these probably in rank order of their distribution in the nervous system. So there's more GABA and glutamate in the nervous system than there are monoaminergic transmitters like serotonin and dopamine. Although someone once told me, and I think it's right, that serotonergic fibers that contain the neurotransmitter serotonin are proximate to every single neuron in the brain. So oftentimes with monoamines, you have a small number of cells, but they reach out and innervate a variety of different targets. Cholinergic neurons are fairly sparse. They're not in the back of the brain. They tend to be sort of in an area that's pretty close to the thalamus. And then peptides, people argue about whether these are neuromodulators or neurotransmitters. They are very complex and produce a number of complex things. And nitric oxide, we actually even have evidence of a gas. It's not nitrous oxide, but nitric oxide. And so gases can actually function as neurotransmitters. And what's interesting about these is that they don't show the directionality of neurotransmitters like this. So they don't start in one place and go to someplace else. They're capable of moving postsynaptically, presynaptically. They're interesting. So because we put so much emphasis on neurotransmitters, we tend to define neurons in terms of what transmitter they use. So we'll talk about GABA neurons or dopamine neurons. But what we've learned, and it's been a recent advancement, is that that's not right. There was this principle in neuroscience called Dale's principle, which said that if you had one neuron, it only had one neurotransmitter in it. We know that's wrong now. And I've illustrated here this sort of presynaptic membrane from a monoaminergic neuron, a neuron that's capable of making, in this instance, dopamine. I did it again. But right next to it is a terminal that is releasing an amino acid like glutamate or GABA. So what we don't have any evidence of is a neurotransmitters mixing in a single release site. But when you think of this neuron with its hundreds of thousands or possibly millions of terminals, it wouldn't be correct to think any longer that each of these terminals is releasing the same neurotransmitter. You can actually have a system like this one where different neurotransmitters are released. So the neurotransmitter receptors, these are the proteins on the backside of the membrane that are translating the effects of the released neurotransmitter. So I've got some things on my screen here that are in the way. But so just to sort of orient you, here's where the neurotransmitter came out. And here on the backside are these receptors, which are on the dendritic spine. So remember, this is the sort of the connection of the pathway. We've got a release site, and then we've got this dendritic spine with the receptors located in the membrane here. And we can, although there are, I think, well, I wasn't even able to get a number of how many receptors there are in the brain, but there are a lot. For example, serotonin, the neurotransmitter serotonin, there are 15 different receptors that serotonin is capable of binding to. And of course, what that means is that a single neurotransmitter can have multiple effects depending on what receptor it binds to. And we can break the receptors down into two broad groups: ionotropic receptors, like the ones that are illustrated here. And this is where the neurotransmitter, again, binding here to the dendrite, binds to an ion channel, which then changes the excitability by moving ions directly through this pore. This is a simple type of ionotropic receptor, and by moving these ions across the cell membrane, charge is moving, and the cell becomes either more or less excitable. They're fast, so the type of neurotransmission that's mediated here is very, very fast. Most of the drugs that we use to treat serious mental illness do not target receptors like this. They target G protein coupled receptors, which are way more complex and way more important for us today. So here's a neurotransmitter binding to this binding site. There's a G protein involved, which sort of translates, comes off of the receptor, comes over to our effector, and produces a change. And this gives us much more, many more options to target drugs. And the majority of drugs that we use to treat serious mental illness are targeting these G protein coupled receptors. They're much slower, as you might imagine, given the number of processes that are between the binding of the neurotransmitter to the receptor and the actual change that occurs in excitability. So they tend to be slow. And by slow, I mean 1 to 2 seconds as opposed to 1 to 2 microseconds. So it's not all that slow. So here we are, we're almost at the end. Let's review this whole thing. So we've got information coming in to the input side of a neuron. Signals are relayed from axons to the dendrites. These dendrites coordinate all of this incoming information and they integrate it. An action potential is generated at the soma. The action potential passes down. The axon invades these axon terminals, causing the release of neurotransmitter at the synapse, and that information is conveyed. So we have a presynaptic neuron and a postsynaptic neuron, depending where it is in this chain. And that's the whole thing. That is basically the process that occurs in all aspects of the neuron to move information from one place to another. And with that background, we can now move to to consider a few topics that I think will be of more maybe practical use to you guys in your role as mental health first aid trainers. And the first of these is addiction. And I'll say that in the course of the last 30 years, we have learned so much more about the neurobiology of addiction. It's really, so much of it has been dependent on the work of this fellow Wolfram Schultz in Germany. We now know that dopamine neurons are intimately involved in reward. And these neurons live in the ventral tegmental area. You may recall the little cartoon we had at the beginning of the talk. And they project to areas like the nucleus accumbens and the prefrontal cortex. And these two areas, the accumbens and the cortex, are intimately involved in drug abuse. What's interesting about the dopamine system is that novel rewards, particularly novel rewards, will increase the activity of dopamine neurons, leading to an increase in dopamine release in areas that these neurons innervate. And when we have an expectation about an outcome and we're disappointed because that outcome is not accomplished, for example, if we were really looking forward to getting an ice cream cone and when we got it, the ice cream was, someone had spilled salt in it inadvertently. These same neurons that would be activated so strongly by the ice cream cone itself are actually made to fall silent, again, as a result of the process that we just sort of went through. And that's meaningful, right? So when these dopamine neurons fall silent, it's a learning signal that the brain uses to avoid whatever circumstance the organism just encountered. So I suppose if there's something to a take-home message from drugs of abuse and what they do to us is that they directly and powerfully activate these dopamine neurons in a way, the same way that an appetitive or an enjoyable stimulus would do. But they do it with no purposeful connection to the behavior other than whatever activities were ongoing when the drugs were administered. And this is a very powerful hijacking, if you will, of the brain's natural reward systems. So I promised Dan that I would explain in a bit of detail the effects of naloxone. We'll turn to that now. So we're going to focus our discussion about drugs of abuse, all of which, by the way, act in the synapses, in the dopamine synapse. So arguably, every drug that is abused is increasing dopamine release from this synapse, either by blocking its reuptake or altering some other aspect of neuronal function. So what we've done here is we're zooming in on this postsynaptic membrane with this receptor located here. And here's our morphine molecule. I'm using that as just as an example. So when morphine is injected, and let's say we've administered it intravenously into the bloodstream, and so it finds its way to the brain where it finds this receptor, and the receptor is there actually to bind to these other substances, these endogenous morphine-like compounds like the enkephalins and endorphins. But morphine is there, and it binds very tightly because the molecule itself, the morphine molecule, is ideally suited to fit into this protein. It fits in there in a very specific way. And when it does that, it causes this G protein coupled moiety that I was telling you about before to physically dislocate from this receptor complex and to go off and do its business. You remember the G protein coupled receptors have the effectors sort of downstream from where the ligand is actually binding, and it inhibits neuronal activity. Morphine always inhibits neuronal activity. Well, I told you that all drugs of abuse excite dopamine neurons. So how does morphine, and if all drugs of abuse involve dopamine neurons, how is it that morphine is addictive? So that happens through a process, and this is exemplary of something it's a concept that occurs all through the nervous system. If you have a drug like morphine that's clearly addictive, and you know that all systems that addictive behaviors involve an increase in dopamine release, how can you take an inhibitory molecule and make it excitatory? Well, you do that through a process that's very common in the nervous system called disinhibition. So here we have this GABA neuron. Remember, GABA is inhibitory. That's its small amino acid. And so it's inhibitory, and it's talking to this dopamine cell, and we'll turn it on. So the GABA neuron is spontaneously active, and it's firing away. And of course, GABA is being released onto the dopamine neuron, which is keeping the activity of the dopamine neuron sort of in check. Now here's the axon from the dopamine neuron. It's going up through the median forebrain bundle into the nucleus accumbens, and dopamine is being released onto a target neuron. And this is that reward behavior that we were talking about. So how is it that we get this reward signal turned on in the case of morphine? So we've administered morphine intravenously. It binds to this receptor site that it shouldn't be binding to. It can, of course, because structurally it's able to. And what it does is it slows that GABA neuron down. So this thing, which was firing pretty fast, and because it has an inhibitory effect on the dopamine neuron, if you slow this neuron down, you speed this one up through a process called disinhibition. You're no longer inhibiting this neuron as much as you did before. And as a result, you get a bunch more dopamine released. And that is exactly how morphine and other mu opiate receptor agonists like fentanyl work. They increase dopamine levels in the nucleus accumbens and other drug-related areas like the prefrontal cortex by augmenting dopamine release through a process of disinhibition. The mu opiate receptors are not on the dopamine cells. They're on these little GABA neurons, and the GABA neurons are involved in this little circuit that is controlling the dopamine cells. And that is what's producing the reward signal. Well, let's go back. So we've administered morphine, and it's had the effect that I've just described in the substantia nigra and ventral tegmental area, and the reward systems have been activated strongly. But there are other mu opiate receptors in the nervous system, and one of the other places that they live in quite an amazing number is illustrated here. Now, our dopamine cells are kind of up here, and we just talked about those, but back down here in this primitive part of the brain is this area, the Bonziger complex, that are involved. These are neurons that project down in the spinal cord, and they innervate the diaphragm and the intercostal muscles that are involved in respiration. And these things are just fire automatically. Many of the neurons in this part of the brain, these are rhythmically active. They control respiration, they control heart rate. And if you record from these neurons, and we can do that, you see that they just have these beautiful oscillatory rates that are tied explicitly to the processes that they're involved in, like respiration. So morphine is on board now, and it finds that mu receptor on this glutamate neuron. And because morphine is always inhibitory, that glutamate neuron is inhibited. And remember, glutamate is a fast neurotransmitter. It's projecting to the cord, to these muscles. And as a result of the slowing down and eventually stopping of these neurons in the pre-Bozinger complex, the neurons that are driving automatic respiration ceases. So we are activating neurons in dopamine neurons, activating neurons involved in the reward pathway, while we're simultaneously inhibiting neurons that drive respiration. And as a result, people get into trouble. So how does naloxone work? Well, here are the two structures. And as you can see, there's an amazing similarity in how they look. They're almost indistinguishable, really, in terms of their structure. So they're very simple or very similar. And they both bind, of course, to mu opiate receptors in the brain. The interesting thing about naloxone is that it's way more potent than morphine. So when it sees that receptor, it tends to kick morphine off. So here we've got naloxone, okay? And if you can imagine, morphine is in this binding site. And now we've administered naloxone, right, to this person who stopped breathing. And it binds tightly to this site, the same site that morphine had bound to. But the difference between morphine and naloxone is that naloxone doesn't allow this G protein to decouple. So this is the thing that I was telling you before about that actually causes the change, right? This is the thing that activates the channel, that causes the inhibition. Antagonists work by binding to the site, displacing morphine or fentanyl or any other mu opiate agonist, but they don't activate the receptor. So as a result, the inhibition doesn't occur. So let's take a look at that. Here we've got this individual with morphine on their glutamate neuron, and respiration has ceased. So we add naloxone, it kicks the morphine off of that receptor. And because it can't activate that G protein, because it can't produce the inhibition any longer, and because it's sitting in the receptor site, morphine can't bind here anymore. Respiration continues, it's restored. And if this process occurs fast enough, we can oftentimes revive people whose respiration has ceased as a result of inadvertent activation of these receptors. So at this point, I want to emphasize something that I think is super important. No one becomes addicted to a drug after one dose. Right, and in fact, there are a lot of people who are making this point, and it's a really salient one and an important one, because it highlights another process in the brain that is key to understanding what causes people to transition from a recreational use into an addicted state. And it's this word plasticity. So repeated administration of drugs produces long-lasting changes in all of these mechanisms that I have described. So we get structural changes, and we'll see this in a minute. We get structural changes in how dendrites look. We get changes in those second messengers that we were talking about before. And we can even affect what transcription factors or what genes get turned on. And this is a slower process that requires repeated administration of drugs that puts people into a state that they don't want to be in. And that's plasticity. The same mechanisms that are involved in learning are really involved in co-opting our brains after chronic administration of drugs. And here's a nice cartoon that I found that sort of summarizes some of the changes that occur in the brain in response to long-term drug abuse. So we know about dendritic spines. We saw those in the early part of the talk. One of the things that the plastic changes that occurs in the brain is the number of spines changes. The morphology of neurons can change. So we know that in some circumstances, drug abuse being one of them, the actual physical morphology of the cell changes. So it can sprout these additional dendrites, which of course, if this happens in regions of the brain, and it does, that are involved in negative valence or fear or deleterious behaviors, this sets people up to really put themselves at risk for developing serious illness. We can change the strength of synaptic input. So that very first cartoon where I showed you those little teeny potentials that get generated when a neurotransmitter hits a receptor on a postsynaptic membrane, and those little small EPSPs and IPSPs converge on the cell body. The magnitude of those changes, the magnitude of those little tiny changes in voltage are regulated tightly in the brain. They can go up and down depending on how often a particular synapse is activated. Tremendous amount of plasticity at the cellular level. And then, as we talked about before, these longer, slower, more adaptive changes, both in gene transcription and translation, but also in these second messenger cascades that lie behind the receptor, which are the targets for drugs that we use to treat the illness. So we can look here at a number of changes in plasticity occur, and will change now from drug abuse to stress, because stress is one of the other things that can really produce some substantial changes in the plasticity of the brain. So 3 areas that are particularly involved in mediating the response of our brain to chronic stress are the amygdala here. That's part of the diencephalon we talked about before, the prefrontal cortex and the hippocampus. So the hippocampus and the prefrontal cortex, the nature of those changes are pretty similar. So we see reductions here. So we see reductions in volume in the cortex. We see neuronal cell loss. There's a loss of dendritic length and branching, and a decrease in spine density in the prefrontal cortex in response to chronic stress. In the hippocampus, we see a similar profile, but interestingly, a reduction in the number of new neurons that are born. So the hippocampus is one of the very few places in the brain where neurons can actually be born, new ones. And in fact, that process, which is called neurogenesis, appears to be an important one in major depressive disorder. The amygdala shows changes that are opposite. This is an area of the brain that, as you know, is involved in fear processing and aversive learning, fear conditioning, all takes place in the amygdala. And here we see a profile that's exactly opposite to what we see in the hippocampus and the prefrontal cortex. There's an increase in the expression of glutamate receptors, which we know are excitatory. We see an increase in dendritic branching and spine density, and a number of epigenetic changes. And these changes, really these plastic changes that occur in our brains, are really what produce serious mental illness. And it's what you guys are seeing when you see maladaptive behavior and you're in the process of deciding yourself whether it's worthy of an intervention. What you're actually seeing is the manifestation of plastic changes in the brain. So talk a little bit very briefly, because we're running out of time, about stress. So most of us would regard stress as a deleterious condition and an important contributor to disease. It is, of course, but it's also very highly conserved because it promotes survival. It's designed to transiently increase vigilance and arousal, and to enhance aversive memories so that we can learn from our mistakes, more or less. And of course, a big part of the stress response is mediated by glucocorticoids, and in humans, cortisol that's released from the adrenal gland as part of the fight or flight response. We also have this elaborate hypothalamus pituitary mechanism for regulating the amount of cortisol that's flushed out. One of the reasons that we know, or one of the reasons that the prefrontal cortex, amygdala, and hippocampus are so intimately involved in all stress-related processes is because they're parts of the brain that have the receptors for cortisol. And when you put a cortisol receptor, like the ones we've been talking about all this early part in the afternoon, if you put a cortisol receptor on a neuron, you make it responsive to stress. And this last point's important. So while these stress-related mechanisms are important for survival, the chronic activation of these systems can lead to anxiety and states of hypervigilance that are maladaptive and can threaten people. I want to distinguish between two different stress mechanisms in the brain that are important for the next few minutes, and we're winding down. There appears to be a mechanism, an ancient mechanism in the brain. It lives back here in the more primitive parts of the brain, the parts that control respiration and heart rate and such, that are receptive to really life-threatening circumstances like hypoxia, the lack of oxygen, or hemorrhage, and these are involved in the sort of extremely primitive, highly conserved mechanisms that are involved in making sure that the organism stays alive. And they involve parts of the brain that are not involved in the processing of emotional stress, which requires an integration with cognitive systems, memory, so when bad experiences can be compared with past experiences, and of course the adding of affect. These stress mechanisms are very, very different than the stress mechanisms that are involved in monitoring the state of the physiological state of the organism, or its introceptive state. And I want to give you a couple of examples of two conditions that appear to involve these circuits differently. So panic disorder, I will say that I don't know whether this remains as vigorous a hypothesis as it had been. But there is this thought that panic disorder involves those introceptive mechanisms, these pathways that are involved in mediating hemorrhage and hypoxia. It's an anxiety syndrome, of course, that is fairly common. It has a lifetime prevalence of about 4%. And its phenomenology is interesting and complex because it includes aspects of an acute fear response, but also an anticipatory fear as well, and chronic anxiety as well. Like all stress-related disorders, there's a huge amount of psychiatric comorbidity, so major depressive disorder, substance abuse, suicide, often associated with panic disorder. But the panic that people with panic disorder experience is a existential panic that seems to involve activation of these introceptive mechanisms, these very primitive mechanisms that are involved in monitoring hemorrhage and hypoxia. And as a result, they involve different parts of the brain than many other types of stress-related disorders. We don't know what the etiology is. It has a very strong hereditability, which is interesting, certainly suggests that, that there is a neurobiological underpinning. And one of the things that makes it, I think, particularly compelling to think that it's these interoceptive stress mechanisms that are involved in panic disorder is that one of the ways that you can really elicit a panic response is by in these individuals is to ask them to breathe a little CO2. So we have sensors in the back of our brain that are CO2 sensitive, and they give, it's called hypercapnia, as you know. It gives you the sensation that you don't have enough oxygen, and it's very reliable that individuals with panic disorder, if they're allowed to breathe even a small concentration of CO2, it will reliably elicit a full-blown panic attack. So it's left many of us with the idea that the brain circuits that are involved in these interoceptive, life-threatening conditions are somehow participating to participating in panic disorder. So post-traumatic stress disorder, a bit different. It involves a couple of different brain areas, the amygdala for sure, which is where emotional memory is stored, and then episodic memory up here in the hippocampus, factual memory. And it's the convergence of this previous emotional memory of a terrifying event, either witnessing it or experiencing it. The emotional load that's associated with that, combined with the memory itself, that elicits and is so characteristic of post-traumatic stress syndrome. There are 2 SSRIs that are approved to treat the illness. Neither one of them are particularly effective. Both are just barely better than placebo. A better outcome, but still not a great one, from trauma-focused psychotherapy. Often these 2 are used in combination. And here's so I promise we really are winding up. I was just absolutely fascinated by a paper that came out about 2-3 weeks ago about MDMA, which is Molly, or it's a drug that made a presence in the rave scene for a few years back. It's actually pretty old. It was synthesized just at the turn of the century by Merck. They shelved it in 1960. It kicked around for quite a while. And the therapist, Alexander Shulgin, as many people in the 70s did, took it and sort of self-experimented with it. He gave it some to his wife, and she was a therapist, and they both really liked it and thought that it would be something that would be useful in therapies. And it kicked around amongst the therapists. I had no knowledge of this. It was extensively used in the 70s and 80s. In psychotherapy for a variety of reasons, for a variety of syndromes. And apparently the people that were using it had tremendous luck with it. Well, word got out that its structure was related to amphetamine and cocaine, and it started showing up at raves, and Reagan criminalized it in 1985. It's a class 1 drug. But this group of therapists, MAPS, some of you may know, a Multidisciplinary Association for Psychedelic Studies, these guys really knew about what Shulgin and his wife had done, and they wanted to see whether they could show whether it actually has any efficacy in PTSD. So the FDA granted a breakthrough therapy designation based on a number of phase 2 studies which showed incredible efficacy in chronic treatment-resistant PTSD. It is a dirty drug. It does a bunch of stuff. So pharmacologists call drugs dirty when they do a bunch of different stuff. And this is an example of one that does. It blocks the reuptake of serotonin in that synapse that we were talking about. It binds directly to postsynaptic receptors. And here's maybe the key: it releases oxytocin, one of those peptide neurotransmitters I was telling you about. And this people call the empathy neurotransmitter. So what it apparently does is that, one of the things that it does is that it blocks emotional memory. And by blocking the emotional component of the experience, it allows therapists to get at what they need to get at to deal with what these individuals have experienced. And I won't go into this more than to say that the treatment response of individuals in this phase 3 study that has recently been published has been nothing short of miraculous. And I think that what is likely to happen is just as we saw with ketamine after the discovery that it was useful in treatment-resistant major depressive disorder, that we are going to start to see MDMA clinics pop up. And I think that could happen within a year. Very quickly, I just want to summarize again that as a neuroscientist, I sort of want to take this on myself, and it's representative for, I think, the entire field. We've learned a lot about the brain, but we have not been very successful in turning our discoveries into real tangible therapeutic approaches. We tend to focus now almost too much on G protein coupled receptors. I tried to point out where the current drug therapy is targeted. Our goal, of course, has always been to reverse the underlying pathological changes that occur in disease, but this is really hard when you're dealing with the plastic changes that occur in the brain. So as we've discussed, it's not just a matter of a receptor being occupied for a few seconds. The brain has this remarkable way of changing itself to accommodate to a new state. And that's really the pushback that the brain gives us in trying to cure a serious mental illness. The reality is our therapeutics, many of them don't cure patients, but they take them to some state where some part of their clinical picture is treated, but it still leaves them with the burden of the unmet therapeutic aspects or unmet therapeutic needs of a disease that's multifaceted. I think personally that this drug facilitated enhancement of structured psychotherapy, it's so novel that it really could be a game changer. And I wanted you guys to know about it because I think it's going to be on the horizon, and it's such good news. I'll leave you with this slide, which will end the talk. There are 3 sources of information that you guys can use to learn more about what we talked about today, and to learn more about some practical aspects of the illnesses that drive the behaviors that you guys seek to detect. So PubMed Central, a remarkable source. All of the articles there are available as free text. You can download a paper on any serious mental illness you'd like, a review paper. You can often, hopefully with some of the information I've given you today, you can maybe even understand a bit more than you would have. At least that's true for some. NIH Reporter, most folks don't know that all of the grants that are funded by the National Institutes of Health are here at NIH Report. You can search by disease, you can research by a clinical trial, you can figure out what the NIH is up to in virtually any project that's ongoing, any funding year, whether it's current. You can find PIs, you can get their email addresses, you can email them for information. It's part of our obligation is to provide information, just like this talk. And then the Society for Neuroscience, which is this amazing organization that I am part of, that has this remarkable resource for learning materials related to all forms of brain-related science, including clinical syndrome. So with that, I'll stop, and thank you all for the attention you managed to deliver.
Dr. Shepard, that was incredible. So interesting. I personally feel as though my own brain has expanded as I've listened and tried to process that really tremendous amount of incredibly useful information. I know we have at least one question in the chat. Leah, can you read that out?
Yeah, so is the addiction circuitry and plasticity the same for alcohol addiction as it is for the other drugs that you were mentioning?
Ooh, what a great question. So, well, two things about that. Of course, there's always plasticity. So yes, but the specific kinds of plasticity that occur, no. And if I've got the slide. So plastic changes, yes. No, there's no question about that. We'll look up here. So these things we haven't talked about, these two things. This is meant to be an ion channel, which is activated by a drug. And alcohol turns out to activate a bunch of these ion channels. And so the adaptive changes that would occur as a result of alcohol would be phenomenal. And the reason is the drug interacts with an extremely powerful way of altering the excitability of this dendritic spine. So I would predict that the plastic changes that occur with alcohol probably occur faster than they do with a drug that's involved with the G protein effect. The second thing I'll say is that there was an open, so there has been at least one open trial. These are clinical trials you don't put much stock in, but there's been at least one, and MDMA has been phenomenally effective. Again, in dealing with alcohol abuse. So my guess is that we won't see an MDMA alcohol clinic in a year, but we will almost certainly start to see phase 2 and phase 3 studies for MDMA use in the treatment of alcohol use disorder. I hope that answered it.
If it did not, please follow up. You can always unmute to ask your questions. Here's another one. Is MDMA and therapy currently accepted as an EBT for HHS funded grant delivery systems?
Ooh, you'll have to, I'm not an acronym expert, so you'll need to uncode the acronym for me.
All right, so the poster, Ruby, if you would come off mute for a minute, or explain in the chat.
So evidence-based treatment, in HHS, they always want to validate treatments as evidence-based for programs that receive funding, like universities. Some universities and hospitals receive funding through HHS for mental health and substance abuse treatment services, but they have to use evidence-based modalities. So I was just wondering if that was one that was accepted, that this MDMA along with therapy.
Ruby, thank you for your question. It's a great one. And while I can't provide you with a definitive answer to it, I'll point to this part of the slide, which is an important component of this study. So the MAPS, the group that has been pushing for the consideration of MDMA, which is currently a class 1 drug, which means it has no therapeutic benefit at all, and is very difficult to work with both experimentally and in humans. The FDA granted this breakthrough therapy designation about 5 or 6 years ago. What that meant is that they were part of this study, the phase 3 clinical trial that was conducted on MDMA, the one that I did such a poor job of describing, was actually done under the auspices of the Food and Drug Administration. So they were literally working over the shoulders of the people that were doing the study, and they had specific criteria, as you know, that are important, like the number of people that are recruited into the study, and a number of other things, how the study was designed, including how the placebo groups were used, because that's always an important part of studies involving drugs with these sorts of properties, is what do you use as a placebo. All of that was run through the FDA. So while I cannot provide a definitive answer to your question, my guess is that they put themselves in the best possible position you could be in to have those hurdles quickly dealt with, because I really do think this is going to be a game changer.
Sounds like it to me. Thank you very much. I think it's going to benefit a lot of the returning veterans who have PTSD as well.
As you know, that's a huge group, and one that even the coldest heart would have some room for. We really owe these people more than we're able to provide at this point.
We've got about 1.5 minutes and 2 more questions. So I think we're going to stop the questions now. Dr. Shepard, in addition to stopping use of a substance, what else can someone do to restore damage done to the brain by years of substance use?
This is going to sound, what you guys already know is no different than what I know. So it's lifestyle changes, right? So and at the top of the list, we talked about the hippocampus, and one of the strange things about the hippocampus is that it's one of those areas of the brain where cells are born. So they seem to start out as part of the lining the blood vessels in the hippocampus, and then through some process we don't understand, they get turned into neurons. And the thing that and if you look at someone who has major depressive disorder, which is a comorbidity for nearly all stress and drug-related behavioral changes, the one thing that you can do to increase the number of cells in your hippocampus is exercise. So if I were to say, if there was a real tangible answer to that, if you could get someone to start exercising, that would likely provide the most direct way of changing the plastic changes that occur in the brain as a result of chronic substance abuse.
Awesome, which dovetails into the last question, which is about dopamine and other non-pharmaceutical methods to enhance dopamine in systems. Do things like exercise, massage therapy, meditation increase dopamine production?
Well, so if you find them enjoyable, yes. I mean, that's really what we learned. And what we've learned is that if you enjoy something, whether it's a video game or that feeling you get when you hit the wall with your tennis shoes on, that releases copious amounts of dopamine. So, if you were to take a neurobiological perspective to changing behavior, think about the dopamine system and think about activities that you can do that will increase the amount of dopamine that's released in your brain, because that's going to have a beneficial effect. It's going to make you happier. It's going to make your brain healthier.
Amazing. Dr. Shepard, thank you so much. This was fascinating, really useful, so helpful. We appreciate your expertise and your time and your support of mental health first aid. So
let me say, it's not having been the beneficiary of your training and how remarkably important the work is that you guys are doing, and how it's changed my perspective about making people aware of saying something and seeing something, and I will be and will continue to be a strong advocate for everything that you guys do. I wish you nothing but the best of luck.
Well, thank you for those incredibly kind words. The perfect close to this half of the program. Dr. Shepard, thank you again. Bob, I'm sure our paths will cross in the future.
I hope so.
Take care of yourself. Participants, thank you for your attention. We're going to take a quick break.
Welcome back.
We got kicked out, Leah. Javon was saying some really good stuff. We had a very good break. You must have saved the best for our group. They were just outstanding. I'm not saying everybody else's breakout wasn't great, but Javon, this guy, I knew it was going to happen. Because once everyone started opening up and we started adding things and started hitting some points, it was time to leave. So, I don't know what Javon is saying.
There was one of our participants was in the middle of saying something during breakout. You mind if I ask her to finish her thought?
Please do.
Jennifer, you were saying that you had been teaching in person in your county. You want to say a little bit more about what that's like, how it felt?
Yeah, so we're one of those counties where the other lady, I don't remember her name, was talking about, but we, the Carroll community college actually does our scheduling and things for us like that. We do like follow-ups to make sure that they're going to come to the class, but we've been doing in person. I think we resumed probably, I want to say like last October or November, and they are just limited. So they limited the class size, and they had all of the, equipment for people to stay sanitary and things like that. So we had 13 people versus we could typically hold about 20 or more in a class. We did have some, of course, die down through COVID where people actually thought the class was going to be virtual just because everything in the world had been turning to virtual, and then didn't realize and wouldn't show up. So that did happen a couple of times. But other than that, it's been picking back up, including two of our actual mental health first aid coordinator is at teaching mental health first aid right now and couldn't be on the summit. So yeah, we've been doing in person, and it's been very good.
Great. Thanks, Jennifer.
Thank you, Jennifer. Thank you, everybody. This concludes the scheduled portion of our summit. Many thanks to all of you for being here, to all of our wonderful presenters, to Leah, to Mary, to Dr. Shepherd. Leah has some networking ideas that she wants to roll out for anybody who wants to stay in the main room after the conclusion of the program.
Thank you, Anne.
Thank you so much, everyone.
Great session, Javon.